So here we have non-visual responses to light exposure in the human brain during the circadian night. This is another paper that Giles van der Waal contributed on. However, he's not the lead researcher this time. This is earlier, much earlier than the other papers that we were looking at. This is 2004. So it's expected that at that time somebody else would have been the lead researcher. Summary. The brain processes light information to visually represent the environment, but also to detect changes in ambient light level. The later information induces non-image forming responses and exerts powerful effects on physiology, such as synchronization of the circadian clock and suppression of melatonin. 1, 2, 3. In rodents, irradiance information is transduced from a discrete subset of photosensitive retinal ganglion cells via the retinal-hypothalamic tract to various hypothalamic and brainstem regulatory structures, including the hypothalamic suprachiasmatic, which we've run into that word before, nuclei, the master circadian pacemaker. 4, 5, and 6. But let's look up that one word. Hypothalamic. Retinohypothalamic. Tract is a photic neural input pathway involved in the circadian rhythms of mammals. The origin of the retinal hypothalamic tract is the intrinsically photosensitive retinal ganglion cells, which contain the photopigment melanospin. So, there you go. It's just saying that it's part of the pathway that controls circadian rhythm in mammals. In humans, light also acutely modulates alertness. 7, 8. But the cerebral correlates of this effect are unknown. We assessed original cerebral blood flow in 13 subjects attending to auditory and visual stimuli in near darkness following light exposures greater than 8,000 lux of different durations: 0.5, 17, 16.5, and 0 minutes during the biological night. The bright broadband polychromatic light suppressed melatonin and enhanced alertness. Functional imaging revealed a large-scale occipito-parietal attention network, including the right intraparietal sulcus, was more active in proportion to the duration of light exposures preceding the scans. Activity in the hypothalamus decreased in proportion to previous illumination. These findings have important implications for understanding the effects of light on human behavior. Results in discussion. Although alerting effects of light have been observed during the biological day, they have been most extensively documented during the biological night, 8-9. We therefore assess the regional cerebral blood flow, RCBF, by positron emission tomography, PET, 12 times at 20-minute intervals between 12:30 and 4:30 a.m. in a group of 13 right-handed 10 male subjects. During the scans, the attention set was kept constant by asking the subjects to count deviant sounds in an auditory oddball paradigm and fixate a red diode. The later light stimulus is unlikely to elicit any non-image forming NIF response because the red light does not fall within the known NIF action spectra, and the luminance level generated by the source is well below the threshold for any known NIF response. All scans were otherwise obtained in darkness. During the scans, the electroencephalogram, EEG, which is, we talked about the brain scan instead of the heart scan, was recorded at FZ, CZ, and PZ, continuously to compute the P300 wave, a positive potential occurring when the expected but unpredictable deviant auditory stimulus is detected. 11. A block of 4 scans was repeated 3 times during the night, and each block scans differed only by the duration, respectively 0.5, 17, 16.5, and 0 minutes. Of immediately preceding exposure to light, broadband polychromatic light, white light, which activates all photoreceptors known to be involved in NIF responses. Our aim was to demonstrate the presence of NIF responses in humans. The identification of the photoreceptors involved in these responses will require further experiments using monochromatic light. Our design ensured that the detected CRCBF changes reflect NIF responses because the alerting effect of light is not time-locked to, but rather outlasts the exposure to light. The NIF response to light was independently assessed by measurement of melatonin plasma levels as well as subjective alertness. Karolinska Sleep Analysis Scale, KSS 12. Sampled at regular intervals throughout the experimental session, figure 1b. As expected, this light exposure regimen decreased melatonin plasma levels. Light exposure also attenuated the decline of alertness that occurs in the course of the biological night, figures 1c. The main analysis of PET data identified the brain areas where the RCBF was significantly modulated by the duration of the light exposure immediately preceding each scan. The simple main effect of the prior exposure to light was estimated according to the general linear model at each voxel, and statistical analysis were computed using a fixed effects model. Linear contrasts were used to identify brain regions where the RCBF was positively modulated by the duration of prior light exposure, as no prior knowledge was available concerning the NIF responses in the human brain. The statistical inferences were assessed conservatively on the whole cerebral volume at the voxel level at p less than 0.05, with a correlation for multiple comparisons based on the theory of random Gaussian fields. Theory of random Gaussian fields. I'm not familiar with this theory. Let's look it up. Gaussian random field involving Gaussian probability density functions of the variables. A one-dimensional GRF Gaussian random field is also called a Gaussian process. And the important special case of GRF is the Gaussian free field. I want to explain why it would be observed in a sleep study. Correlation. I think it's looking at the the values that are generated using their scan, and they're taking them and they're using that to look for a Gaussian distribution. And so the central limit theorem dictates that at any point the sum of these individual plane wave will contribute to and exhibit a Gaussian distribution. So they're looking for that. That's the only thing I can think of, because why they would be using a Gaussian field or this theory. Several cortical areas were more active in proportion to the previous light exposure. The striate cortex, the right intraparietal sulcus (IPS), the bilaterally and extrinsec area (Figure 2 in Table 1). The regional distribution of these robust effects suggests the light-induced recruitment of a set of cortical areas involved in attention and behavioral and neurophysical logical data confirm the successful recruitment of attention by the subjects during the scans. The performance of the auditory task was near maximal under all conditions. 0.1 error per scan. Likewise, the P300 amplitude and latency did not significantly vary along the night or the light exposure condition. P was greater than 0.05. The parietal cortex, especially on the right side, has long been implicated in attention. 13-14. The right parietal lobe has been shown to be activated in response to auditory attention tasks. It is activated when subjects are asked to attend to sound, pitch, or spatial localization. 15-17. The parietal areas involved in visual attention overlap with those participating in auditory attention. 17. During visual attention tasks, functional imaging studies often report activation of the superior parietal lobe, or less consistently in the inferior parietal lobe. 18. More recently, it was shown that several sites in the interparietal sulcus are specifically activated when the subject has to direct and maintain his visual attention on a specified location, a condition that corresponds to our experimental situation. It seems likely that increased activity in the IPS in our experiment reflects the light-induced modulation of attention directed to both visual and auditory stimuli. As the same low luminance visual stimulus was present in all scans, the change in activity in striate and extrastriate regions cannot be explained by any immediate change in visual input. A top-down influence from the parietal regions onto the occipital cortices appears more likely. It is well known that attention can enhance the baseline activity and neural response to visual stimuli in the striate and extrastriate areas. 18. In non-human primates, several areas in the IPS do not project to visual areas. 20 and 21. The top-down effect of IPS on early visual areas is further supported by the results of physiological interaction. This analysis identified changes in functional connectivity and that were modulated by the duration of light exposure before each scan. 22. There is a significant interaction between measured activity in the partial region and light exposure when trying to explain activity in early visual areas. This can be interpreted as an increase in the effect of top-down parietal influences on visual cortical activity, which depends on light exposure. No change in RCBF in relation to auditory task was observed in temporal areas. However, in contrast to occipital areas in visual attention tasks, the auditory areas in the temporal lobe are not systematically activated by auditory attention. 16-17. Consequently, our current data do not rule out a modulation of auditory areas by higher order parietal areas. An alternative hypothesis of this cortical activation pattern would be that visibility of the dim spot varies over the period of dark adaptation. This alternative explanation seems unlikely. First, the reversal of dark adaptation is a very rapid process. If dark adaptation were the main mechanism underlying these effects, the main difference in RCBF would be between the light conditions on the one hand and the long dark condition on the other hand. The activation pattern during the 3 scans immediately after light exposure, scans 1, 2, and 3, would be similar and significantly differ from the brain response observed in the scan following the longer period of darkness, scan 4. It is not consistent with our results. Second, the red diode was fixated. This implies that light was falling on the foeva. Dark adaptation for centrally fixated fields is minimal. 23. Third, the diode was red. Variation in threshold luminance is minimal for longer wavelengths. 24, 25. Although these considerations strengthen the interpretation that the observed effects reflect changes in attention mediated by non-classical visual mechanisms. Future experiments in which the attention set consists exclusively of auditory stimuli are needed to demonstrate empirically that variation in visual sensitivity indeed does not contribute to the observed effects. In rodents, one of the best known irradiance detection responses is a change in firing rate in SCN neurons. 26. The firing rate of SCN neurons typically increases during light exposure, but shows a substantial prolonged, i.e. several minutes, undershoot when the light is switched off. Figure 5 and 26. And the firing rate of hypothalamic neurons outside the SCN can be suppressed by the light H. Cooper personal communication. The responsiveness of SCN neurons is maximal during nighttime. In the present study, most of the scans started nearly immediately after the light was switched off. Figure 1. Linear contrasts were used to identify brain regions where the RCBF is negatively modulated by the duration of prior light exposure. A significant decrease in the RCBF at switched off was found in the suprachiasmatic region. Peak voxel equals 80 minus 10. Z equals 3.27, PSVC equals 0.02, figure 3. The spatial resolution of PET scanning does not allow us to specify the nuclei included in the deactivated area. We surmise that the later correspond to the SCN and other hypothalamic structures involved in NIF responses in rodents, such as the subparaventricular zone or the ventrolateral preoptic area. 6. How the irradiance information detected by the early NIF system eventually modulates cortical activity cannot be derived from our data. Subcortical activating structures, such as the locus coeruleus or the histaminergic neurons or the posterolateral hypothalamus 28 are involved in direct and indirect pathways are likely candidates for modulatory structures that indirectly receive the irradiance information. Given the widespread influence of these structures on the whole forebrain, it is likely that light globally enhances the responsiveness of cortical neural populations, thereby facilitating the processing of external as well as internal inputs. As the subjects were engaged in an intentional task, the regional activity pattern reported here reflects such as a task-independent enhancement of cortical response rather than a specific effect of light irradiance information on parietal and occipital areas. Conclusions. Our results demonstrate the NIF responses and humans extend to modulation of a large-scale network of cortical areas involved in attention. These data have important implications for the understanding of the effects of light on human cognition and the design of novel lighting environments to improve alertness and performance. And then it gets into the procedures, the subjects, and references. So, this study is a precursor to all the stuff that we've been talking about, and so they're just starting to realize that light has non-visual responses to light exposure, and how it affects the circadian rhythm. Not even just seeing, not non-visual responses to light. And some of the wavelengths that are on the non-visual, which is interesting. But I mean, this is before though they were looking at blue light and what which was the more effective light. This is more like types of light that are you're not even able to necessarily to see. That's such a decreases along the night. So it's definitely interesting. And you see how it affected his later work. So it's you're looking at almost like a chain of causality. Like this led to other things. And granted, it's not as informative as the other stuff because the other stuff is more on the topic of what you're interested in. This is more of a precursor that is leading to all that. But it still should be of use because it's talking about light and circadian rhythm and circadian night and how the human brain reacts to light. So it might have been a good one to start with if we had done this in a chronological order. And that might be something to think about for future ones, is to add it in a chronological order. Alright, well, that's it.